Good afternoon, everyone. I'm Nicole Lance, and I'm the business manager for the CBSR. And I'm going to do a brief intro into iLab, and then each of the cores will do a portion of the demo. So getting started with iLab, we have a landing page, which gets you in. You can see the cores and a brief description. Core contacts are myself and Amy. And the CBSR has an email address as well, which is monitored by Amy and myself. iLab loves Google Chrome or Mozilla, so if you have any errors, just check that you're using a browser that it loves versus Explorer or another one. So from the landing page, you have a core facilities and sign in or log in. Core facilities page gives you all of the CBSR codes. You can get a about and brief information and contact information. Sign up is basically step by step. It'll get you through to your core of choice, and then from there you can request access to the other cores. So once you have an account, if you do not see the schedule equipment or request services tabs, please contact Amy or myself, and we will do some behind the scenes stuff and get you access to those. So request services tab is kind of where you'll locate scheduling consults, training, requesting services if you would like the core to do them. It's a little bit different for each of the cores, but that's basically where those items are located. View my requests. This will show you all of your requests across all the cores and status of their progress if the core is doing them for you. So the hamburger tab, you can get to most of the same places. Under my cores will be the cores that you have access to. Again, you can view your requests from here and your reservations across all the cores, so current and past reservations. So for the PIs and managers, you can see all of your people's stuff as well under my reservations and requests. And you can also see invoices. Under the manage my groups, you can access your members and chart strings. You can enter chart strings yourself, or you can send them to Amy and myself, and we can do that for you. And chart strings, because iLab is integrated with PeopleSoft, it does require, if you have a project, that you would include the analysis type, which is usually a GLE, but for some departments it could be CGE. So if you don't know, that would be a good question for either your PI directly or your business manager. Calendar reservations. So Flow and MIC have calendar reservations. You can edit the time that you have used, whether you've used more or less, and you can migrate to your reservations, open your calendar reservation, and then the little pencil guy and save. You will only be billed for the time that you actually logged. You won't be billed for the time that you reserved. So again, if you need any iLab help, Amy and I can assist you. And if you're a do-it-yourselfer, iLab has plenty of useful tools under their help tab. And next up is Flow.
Thank you. Hello, and also the guys if there are somebody connected, thank you so much. My name is Roxana Del Rio Guerra. I go for Roxana, she, her. I'm the director. I'm the director of the flow cytometry and small particle detection facility. Welcome everybody. One thing is to have an iLab account, and another thing is to be user of a facility. Then to be user of my facility, you go to iLab and you ask for a request consultation, or you type my name, Roxana, and the only Roxana at UVM, and then we provide the email, and then contact me asking that you would like to have a consultation. Okay, and then once we have the consultation, we will figure out what do you want to do. Regardless of what do you want to do at the facility, all new users will receive the policies and procedures, including the data management plan. All facilities has such. All users will be updated from time to time when we have updates of the policies and procedures. In addition, you as a new user will need to sign an agreement. This agreement, regardless of the time that you sign, will be updated, will be renewed every fiscal year. We need to sign by you as a user, as co-signed by your PI. And this is just a gentle reminder of your commitment that you have been establishing with our facility to basically follow the policies and procedures, to cite the work that you have done by yourself or by staff in any publication, paper, presentation, poster, etc. And every year, you will need to provide that information to us, and all facilities are very important for the fitness of the research at UVM to have accountable for research that has been done in facilities. Please, this is a very important stuff for us. If you are independent user, that's meaning you want to use one of our 4 analyzers, then you will need to be up to date to the biosafety transcript, name BSL2, as well as the OSHA blood-borne pathogen courses. The first one is every 3 years, the second is every year. Then if you are not up to date, your access to the facility will be denied. I will send you a reminder, but please, this is something that you need to be checked on. If you are users who do not work by yourself in an instrument, that's meaning you require staff-assisted services such as run the samples for you or do flow cytometry, run your cell sorting, you don't need to have those biosafety plans. If you want to use the facility to analyze by yourself, then there are some steps, easy steps to go. First, you will need to submit a consultation request. This is a platform to know each other and to know what is your project. It will be assigned an instrument for your application. Then after that, once you know which instrument is more like to use, better for you, you will submit a training request. Then we will set up the day, the time that will accommodate for both of us. You as a user will need to provide the controls and the samples for your training. And then at the beginning, the first couple of sessions, you will need to book with me to run together your samples. Once you are comfortable running with the minimal requirement to run by yourself, then you will have up the fingers, and then you will be added into the system for your own reservation, okay? And you will be called independent users. You can also provide samples to us, and we will run for you. In that case, you will need to submit a sample submission request via iLab, and it's very important that you indicate the day and time of the drop-off sample. That doesn't mean that the sample will be run that day, that time. That will allow us to organize our schedule to provide a time to run your sample, okay? Then for clarity on that, here is where you will need to find the time and the day in which the samples will be dropped off. And next, you will need to fill out this form with some useful information for us to run your sample, and then you will upload. If you have questions on that, please send me an email. I'm very happy to reply. Usually allows me 48 business hours to reply your email. If I don't reply in 48 business hours, doesn't count the weekends or the holidays. Please send me a reminder. I don't mind. Sometimes either going to the others' emails to me or goes, I don't know why, into my spam. Okay? I don't feel offended if you send me a reminder. If you want to do a cell sorting at a fax, fluorescent activated cell sorting, staff is the only personnel who is using the instrument. That's meaning we do not provide training on this instrument. You will need to submit a sole request, allowing 48-hour business hour for confirmation or reschedule. You will provide 2 tentative days and time that will work for you the most, and you will need to wait to receive a confirmation. Remember, the fact that you submit a request form doesn't mean that the day time that you put on will be the day for your sorting, okay? I am we don't have a delay on sorting, no more than 48 hours for delay. I'm very good. And sometimes people send me an email today and say, hey, Roxana, I'm growing my samples. They are really ready. Do you have time tomorrow? And I don't mind to accommodate, okay? But you need to let me know. The fact that you submit via iLab doesn't guarantee that whatever you submit at the daytime will be processed. Then you will need to fill out some important information, such as if it's human cells, you will need to provide IBC protocol if you have a DNA or you have some pathogen, etc. And you will need to provide day and time of potential sorting. Then here, you will submit, I need some information from your sample that will be important for me to set up the instrument for your application. If there is any question on that form, I will contact you to ask for clarification. If you really don't know how to read or fill out, send me an email and I will help. You will upload, somebody removed my stuff. Then something that I wanted to put here is that once a service is submitted, all conversations from that service will be via iLab. There is in my past, I was having something indicating where to submit. I hope somebody else is putting how to submit some questions, and please do not contact me in my personal email, UVM email, regarding to a service that is already submitted. Because imagine that I need to combine multiple platforms to figure out what we say about your service. If I need to send documents for you, PDF or anything, there is a button that I will send you. If there is some important information that I want you to know in the 3 about our core schedule equipment and request service, there will be a big 24 font bold red that I will say what I need, okay? For example, right now I already telling you that I will be off office on October and November and December in order for you to figure out how to manage those times. If you need a staff service, assisted service, those times, the facility for those type of services will be off. You will not able to submit requests during my absence, and please do not send emails via iLab because they get lost, okay? Remember, if you have any question, contact me. We are very happy to help you and guide you because, believe it or not, iLab is not as friendly as we want. Thank you.
Hello, I'm Nicole Bouchard. I am the lab manager of the Microscopy Imaging Center. So today we're going to just talk about some of the things that are different for iLab from the perspective of MIC. As Nicole showed you, really your best friend when dealing with MIC is the Request Services tab, that blue arrow on the top. This takes you to the page that shows you pretty much everything that you would want to do to initiate any kind of contact with MIC. The two main ones when you're first getting started are that you're going to need to fill out a new user request form, and you're also going to need to fill out a biosafety assessment. These are our protocols that will help us decide if you've done all the proper safety training. It's going to ask you to upload all of your safety training from both AHS and the size shield, and any of the city trainings that might be relevant to the work that you're doing. It's not above and beyond anything that you're required to do as an employee at UVM. It's that basic training that you're supposed to do within a few months of being hired. If you intend to use an instrument that has any laser capabilities in MIC, then you'll also need to do the laser safety training and add it to your training transcript. And then you're just saying that, yes, I've agreed to participate in all the training that is necessary. You're also going to have to sign the bottom of the document. We don't have the policy of updating this every 12 months right now. It is something like Roxana is doing, but that is something that is in discussions as to what we might want to do. The important thing is at the bottom here, you're looking for the chart string that's associated with the work. In this form, it's not that relevant because it's just the user profile form, but you do want to get in the habit of looking to see what chart string is being associated with any reservation that you're making in iLab. A lot of PIs have multiple projects going, and there'll actually be a drop-down menu here that will show you 3 or 4 different chart strings sometimes. And we don't always know what project your chart string goes with. That's pretty personal to the PI. So just make sure that you double check with the PI that you're using the chart string that they expect you to be using. Biosafety assessment, this we want to know about the samples that you're going to bring into the core facility. Have they been vetted with an IBC protocol? If you answer yes to that first question, you're going to be prompted to enter your IBC protocol number. So please contact your PI to make sure that you know what those numbers are and be able to provide them. And just basic information about how the sample has been fixed and that you agree to take all the proper precautions to deal with the sample that you're bringing into MIC. This form will also flag MIC staff. If you answer certain ways to this, then we will review the form, and sometimes we might reach out and ask you to contact your EHS representative to just kind of go over what you intend to do in MIC, make sure that they're comfortable with how you might be bringing the samples into the facility and how things might be handled while you're there. We just get a validation email really from the EHS staff that they're comfortable with what you're proposing to do. Then moving on to training requests. So you probably are aware the MIC has about 25 different instrumentation available in the facility. There's a wide range of things that these instruments are capable of doing. Anywhere from basic light microscopy, microtomy, up to electron microscopy. And you can request training on any one of those instruments. The trainings are really geared toward what your immediate need is. As you might imagine, there's a wide array of possibilities there, but even within one instrument, they're capable of lots of different things. So we really try to gear the training toward what you need in that exact moment. So we ask you to bring a sample with you when you do the training. We also will start with a control sample that we have provided, just so you can see what the imaging should look like in an ideal situation. And then we quickly move on to the sample that you have brought so that we can gear everything toward what your need is. Keep in mind that, as I said, these instruments have a lot of capabilities. So if your needs change, please contact us again. We're happy to sit down with you and discuss how your needs have changed and help you gear your imaging protocols to the new needs that you're putting forward. At the bottom there, I've also highlighted that, if you have any questions, concerns, if you're coming here from another university and you have 30 years of confocal experiments, that's helpful for me to know, because I can say, all right, there's a lot of this that I don't have to drill down with you, and we can kind of shorten or accommodate the training time according to what your level of experience actually is. Then once you're through all these wonderful forms, and maybe you don't want to do the work yourself, maybe you want MIC staff to do it, then you're going to reach down to number 5 here, which is a work request. And when we want to fill out a work request, it's going to be that we want MIC to do something for us. Our staff, Kira is here, and we also have Brad and Jamie, so one of the 4 of us will be able to see what it is that you want to do, and we can do that work for you. One really important piece that we've added to this work request is an acknowledgements validation piece. We really need you to acknowledge that you've done this work in MIC. This is how we continue to be funded and supported by the Dean's Office. They want us to track publications. They want us to track every interaction that you have. So if you can acknowledge us in your presentations, in your manuscripts, in your grants, in this format, that really helps us to continue to exist. So the work request, in MIC, it's pretty open-ended, right? There's a lot of things that you can do with the instrumentation there. So our work request is pretty much a paragraph. Tell me what you want to do, and we will figure out what the right technician is to help you. Maybe we need to set up a consult to talk about the details of this further. If you put something in there that's that I have a lot of questions about, I might respond to you and say, hey, let's talk about this before we get too far. And in the bottom there, this is something new. We've been asked to also keep track of IRB and IACUC protocol numbers that are relevant to the work that you want to do in the lab. You have to do nothing other than provide me with the number for whatever the materials are. It's just come up a few times that maybe those numbers were not kind of in place the way that they should be. So they've asked us to be a space where we can kind of filter through that. Consult requests. So this is, if you have a project that that we need to talk about, go to that consult request tab. Same thing, give us an idea of what you want to do for this project, and we'll pull together the proper people to talk about what your needs are and how we might be able to help. That was in the wrong order. And then kind of what Roxana was saying. So at the bottom of these requests, you'll have this little add comment piece up there. Once a request is active, we like to keep a running tab of what the communication is regarding that project right in iLab. In MIC, we have 4 staff members. We often tag team these projects, passing things back and forth, try to get it done as efficiently as possible for you. If someone has the time to do some sectioning or some staining or imaging, we like to try to make it as efficient as possible. And it's super helpful for us internally to have a running dialogue of where the project is at any time. And the investigator gets email notification, should they choose to, when there's a comment added here, so that you can see in real time what's happening with your project. And then a little further on there, that other blue arrow points to the add attachments. So any data that's generated that's not huge imaging data, say like a protocol sheet or an Excel file, or maybe we pulled a couple of slides together of images that were representative from your experiment, then we'll attach it to the document here, so everything kind of stays together, and you have an idea of what we're doing for you. And that is proteomics.
Hello, how are you? My name is iLab with the proteomics facility. So for those of you who don't know me, I'm in the facility. You can just walk right like down the hallway. We're like all the genomic facility, full cytomics facility, and my colleagues are to the left, but we are to the right. So feel free to ring the wall. Feel free to ring the doorbell. We have doorbell just recently installed. So thank you so much. Our facility is supported by Vermont Biomedical Research Network. VBRN is an INFI grant funded by the Vermont Biomedical Research Network for the past 20 years. So again, as my colleague in other facilities, we usually begin a project with a consultation, right? As Nicole and Nicole mentioned that you can submit 2 requests in iLab, one for consultation and one for surveys, and that's it. It's supposed to be simple and straightforward, and believe me, it shouldn't be complicated. So, so we request investigators before the consultation. We request the investigators to submit a consultation request. So for the consultation request, we would love to hear more about your project. So feel free to put in as much information as you need. The reason is that after many investigators told us that okay, after the graduate student left like 2 years ago, I want to get the data, but I checked and no program, no idea what they wrote. But it's not my fault, right? So, but it turns out that we will keep everything in iLab. It is a little bit. It is relatively easy for us and for the PI to go back, according to the iLab number in the lab notebook to check all the work we have been done for you and for the lab members. So again, as you can see, and please, while you're filling out the information, please save the button, please press the save button. We don't want you to lose the information while we're typing in the information, lose everything. So another thing to say is that you can see some of these items are marked with the asterisk. It means that if you don't fill out the information, the form cannot be submitted. So it can be a frustration generating process, while some of the investigators in the past, I cannot get the form submitted. What's the reason? One of the big reasons was because some of these item was not filled out properly. So so that's it. So once you once you hit the submit button, your lab and the facility colleague in the facility should receive an email confirming that the consultation is has been submitted, so we can start arranging a consultation within the next few days or the following week. But during the meantime, you may think, oh, I'm sick, I don't want to cancel the consultation, what should I do? So actually, after you submitted the request, you can go in to add comments saying, okay, I cannot make it this week, and we rearrange the consultation to the week after. So you can do that too. That's one of the features, a couple of features that we like in the facilities. One is the add comment, and the other one is the add attachment. So when we use the add attachment, you can actually upload some of your publication from your lab, to the iLab. So me and my colleague will try our best to read the publication you uploaded before our meeting. So most of the time to understand more about your project backgrounds. And during the consultation, we usually go over a couple of things. First of all, we go over the project background. You guys are biologists. We are a kind of technical person. We want to learn from you. And also we want to understand, your more like biological question. Another thing is that we'll go over what kind of proteomics strategy can be offered to you. And the third is that, what we can realistically expect from us and what we can realistically deliver. So a kind of manage expectation. So, and during the meantime, we can also on the facility side, personnel side, we can upload some of the paper that describe the experimental procedure, and also we'll upload code in Excel. So, and we'll talk about the code while we're during the consultation. So we would like the PI to be present in the first consultation so that we can approve all the charge about how much it actually costs. Sometimes we work with graduates, say, okay, we'll do one particular study, a big study, no problem. And then later after we finish study together, oh, how come it's so expensive? So we want to make sure that from the first consultation, everybody's on the same page. So this is one of the example of the code in Excel. It lists all the pricing structure. There's the UVM standard way or the external way. So again, some of these experiments can be expensive, but it doesn't necessarily need to be the case. During the consultation, we kind of see, okay, do you really need to do this experiment on that level, that quantitative, that comprehensive? Most of the time, some investigators, I don't need to identify like a 7,000 phosphohectare. We can just do a little bit, which I don't have an undergraduate, they don't need like 100 target in order to graduate. They just need one protein to do the mechanistic study. So some of this pricing is very variable. So it would love for you guys to contact us. Don't be intimidated by the pricing on their website. So again, for after the consultation, some investigator will go, oh, maybe I go at home to think a little bit, maybe I don't want to do proteomics. But for most investigators, after the consultation, they feel extremely excited. They want to get this experiment started right away. If that's the case, what we want to do is that you go to the second request, we will have you to submit the service request. So this is first service request. Unfortunately, what you see the first thing is not to ask service, is to ask you to acknowledge our grant number, because it's very important, right? Because besides the income expense, our facilities almost like 7 to 80% is supported by Vermont Biomedical Research Network. When every year the external advisory committee come here, they don't see, we prepare this type of report, they don't see most of it. They only go to the last page to see how many publications actually cite our facility to demonstrate our expertise at the University of Vermont. So again, you can see all this intimidating web asterisk. It means that you have to really need to sign it and pay it and agree that you need to study our facility. But of course, there's a whole page of this kind of thing. You don't need to read this fine print. Only thing you need to do is just like simply state in your manuscript that okay, the work is supported by BBI proteomic facilities, cite our grant number and the RRID, and that's pretty much it. So again, some usual stuff, we want to know a little bit more from you. This like for grant checking, for this like the data tracking purposes. And this all the name, you can fill in whatever you want. And we found this to be very useful. Sometimes our graduate students put in a specific log number where they actually put that number into the lab log. So in the future, even like, we cannot elucidate what they wrote in the lab log. They have that number. We can really go back to the iLab to elucidate what we have done over the course of like 3 or 4 months work. And again, we ask you to upload and gel image you have. Some of this gel image allow us to assess the sample quality. And of course, we need to know the sample number, we need to know the sample name and how that name is associated with the sample, the sample description. So okay, finally, adding services, we have put together a little instruction how to do it. It's to be very simple, but one of the most frustrating things users find is that we really cannot find the add service button. The font size is extremely small. But what we show here is that but you will find it if you are patient enough. It's basically under please save your form. You just click this button and you basically populate a bunch of services. And if you remember a couple of minutes ago, I showed you an Excel spreadsheet where you can basically easily go into each of like the category the services belongs to. For example, in this case, LCMS, you just go into LCM, you find these services, you add in this number of samples you want to analyze, and the iLab actually automatically calculates the subtotal for you. So for some of this complicated study, that's 5 or 6 items belong to a certain study, so it might take some time for you to enter this number. But if you find difficulty filling the form, feel free to reach out to me or my colleague. Will just Zoom with you and get it submitted right away. Just like test our memory, just remember this number, 4,984. So if everything goes smoothly, I should say congratulations. So the number, the total should be 4,984. So exactly the same, you have done nothing wrong, and then you submit the congratulations. And the future, so all the communication from this point on is full iLab, all the result we send the result full iLab, and then we'll let you know, okay, we have uploaded the result. Of course, in the reality, we're going to have a lot of like explanation and details about the data. So I think that's it. Thank you so much.
Who's going to go first there? All right, so my name is Scott Todd. I'm the technical director of the genomics facility. I've been here for about 25 years. I just found out I was eligible for a rocking chair. Anyways, some of the stuff that has gone over applies to us as well. So I don't want to go over every. Oh, these are yours too.
No, it's just
Okay, cool. Okay. So we're a little bit different. So most of what you've been hearing are cell-based assays. Microscopy imaging does cell-based assays. Roxana does cell-based assays. We're genomics, we're core, you submit samples to us, usually DNA or RNA. So safety training isn't something that we have to worry about because we're handling nucleic acids when you bring them into the lab with the freezer. However, one of the things that we do have is consults, and that's been gone over a few times, and we do want consults for large projects. So same way you go into proteomics or microscopy imaging, we have the same tabs that they have. So you can actually schedule a consult. We usually pull together a Zoom meeting. So we can all get together and talk about it because a lot of these projects get pretty expensive, not unlike proteomics. We can get your projects at their single cell projects up to $20,000 or more. So we want to have these consults with you. Even if you think you might know exactly what you're doing, there might be some facets that you don't know about. So this is a very clear image here, but we have our work requests that can be done in two ways. One is we have a genomics work request. This is where we bundle a whole bunch of services together. So if you're doing single cell analysis with us on 10x, you might have a DNA extractions, you might have DNA quantification, you might have RNA analysis by bioanalyzer. There's a lot of pieces and parts, and you can have sometimes 20 or 30 services go under what we call a work request, and that is right up here. So we also have non-bundled. You can select them one by one. And if you look, sorry, this is not very clear, but we have a large menu here. This says 10x, bioinformatics, shared resources, and digital droplet, qPCR, general genomic services, nanopore sequencing, and next-gen sequencing. Next-gen sequencing, although nanopore is next-gen sequencing, we separate out because nanopore is pretty specialized, and actually now it's our most popular assay in the lab. This is just an expanded version that you're seeing down here of what's happening in Nanopore. There's about 25 services on Nanopore. So each one of these headings, if you see the drop-down bar, there's sometimes 25, 30, 40 services that we offer. In the whole lab, I think we like have 121 services. So it's very different. And it's you submit a sample, you buy one item, and you get a result back. So one of the things when you fill out iLab, we do like to have up here where there's this box that says labels. So this is something you guys can fill out. I just put in mouse clean study up here, but if you have the name of the study, much like what I just said, is just go ahead and put your study name up there or how you can identify these samples verbally, not necessarily samples number, because one of them, you might have 45 samples, or in the case of somebody just submitted 672 samples to us, so you're not going to go by the sample number. But this is just qubit quantification. Don't forget when you put out 6 here, like I just said, it should total out to 2604. And if you didn't put it in and hit enter, it won't upload that. And then down below, this is where you would enter your sample data, like samples 234, 344, blah, blah. And then you can upload right next to it. Under here should be Excel, but you can upload an Excel form. For example, we do quantification in 96 volt plates. You have 96 samples that have already been filled out at your leisure in Excel. You can upload them to us. So you don't have to fill out our form. It can be automatically uploaded. So data attachments, more done doing things, qubits and qPCR and stuff. Down here, there's a little part that says attachments. What I've said is about as small as it can actually get. So this is where the data can be found when we're done if it's small data. This is only good to, I think, 30 to 50 megabytes. After that, it has to be done by file transfer or it goes to the Vermont Advanced Computing Center, which is where a lot of our stuff lies. So a couple of things I want to go over for the lab. So a lot of people come to us with the same issues. And I'd like to, so we have technical consults. So we really encourage those, as I just explained. They're required for like GeoMax studies and 10x studies. We're going to require you to sit down with us because these are really expensive. But if you're just wondering about some project that you're not sure, even if it's just troubleshooting PCR, you're welcome to send us a note. We're happy to help you go through PCR and help you design and figure out primers, but it's not required. It's just something we want to help you with. And then sample submission, just so you know, when samples come in, they get placed in our -20 freezer. When you come down into the facility, it's when you walk down the hall and you have to ring the doorbell. We're more than happy to get the door for you. We want you to come in. They just have us locked off. Seems like all of UVM is locked off now. But when you come down to the genomics facility, there's a freezer right there. So they all go in there and it's nicely thought out. Thank you, Stacey. They got it all organized. Thawing freezers is something we do a lot of, right? And cubic and bioanalyzers are just placed on the shelf and it's all labeled in racks. They say DNA or RNA or qubit. So iLabs, please fill out iLabs before you bring the samples down because we still have samples that we've done that nobody's ever submitted an iLabs for. And we want to make sure we get your data as fast as possible because we want to sit around for 3 days waiting to do it. But sometimes people go, I'm going upstairs right now to fill out my iLabs, and then they don't. Now we're sitting there going, where does this data go? So please do that. So samples are submitted in 0.5 mil and 1.5 or 96 well plates. Please don't come to our lab with 0.2 mil tubes. There are contamination nightmares on every level. We don't even have 0.2 mil thermocyclers in our lab because of contamination issues. So qubit samples 3 microliters, bioanalyzer 2 microliters. Just make sure you do that. A lot of people give us their stocks. Please don't give us your stock because we throw them away and for small things like quantification. Then labeling our tubes is our biggest issue in the facility. You can imagine we get probably 5,000 tubes a year down there. And we would like to see a 5-digit label. 4 would be the minimum. Please don't bring tubes that say 1, 2, 3, 4, 5. Unless you really need to, but it's not going to be fun because we can't tell the difference between a Z and a 2 and an L and a 1 and an S, and you know you can see how it can get tough. And everybody wants to submit things 1, 2, 3, 4, 5. So if Sal comes down and you got someone who's 1, 2, 3, 4, 5, and then Y comes down 1, 2, 3, 4, 5, it's like I don't know who's if they fall. You can't sort of. So please don't do that. This is how we want them labeled. You don't have to do it by typing it right with a Sharpie, but you're labeled DNA. I want to know what it is. Don't DNA. 23 nanogram per microliters in that date, and put it as much as you can on the top too. Don't kill yourself to do it, but make sure the sample number's up there and the quantification. We'd like to see the quantification. So quality control for sample submissions. So when you submit samples, you got to quantify them by qubit. I assume many of you have a qubit. Make sure that you qubit them, and we're not relying on nanodrop. If you can't do it, we'll do it for you. We have a service for qubit spectrophotometric analysis. Likewise, RNA quality should be analyzed before we move on to next-gen sequencing or nanopore sequencing or cDNA synthesis. We need to see that your RNA is pretty good. So we will upload this to your iLabs when you select that. And NGS libraries, if you build your own libraries, for example, Seth makes a lot of libraries. We need to see the bioanalyzer size assessment just to make sure that the insert size isn't going to gromp the sequencer when it goes to the sequencer. And then one thing that I want to encourage you is that if you have to do dilutions of 200 samples before you bring them in the lab, this is not okay. I did my dilutions of 5 microliters and now they're all 5 microliters. It is totally impossible for you to dilute 10 samples to exactly 5 nanograms. So this is right. So when people when they dilute their samples and re-quantify, it should look like this, 4.5, 6.1. We're okay with that. But when it says 5, we don't even know what it really is. And that can be a problem for your final data set. Little policy on storage. Don't submit your stocks. Please don't give us the only thing you got. Because if a freezer goes down or we throw it away, it's gonna be not good. And make sure there'll be a box with your name on it, the -20 or -80, depending on how they need to be stored. And we'll give you a box if you don't have one. But I will tell you, after 6 months, if they're in our freezer, we reserve the right to discard them. So if it's 5 months and 29 days, you got a day. If it's 6 months and a day, we might throw them all away. So just keep in mind, and we've had some problems there where people think we're a storage facility. We still have shared equipment down there. We have a NanoDrop ND1000. So if you want to do 260, 280, 230, or just get some rough estimates, the NanoDrop's down there. It's free. We have a ChemiDoc system, XRS, that's still down there. It's a beautiful machine. It's like 7 years old now, but it works perfectly. And those are done by sign up on our shared, I think it's under General Genomic Services or the ChemiDoc system. So bioanalyzer, we usually turn those samples around 24 hours. RT-qPCR digital PCR is 5 to 10 business days on that. The singular G4 next-gen sequencer, or Illumina sequencing, we'll call it that, is 5 to 10 days, but it really depends on how many samples you come to us with. 45 samples for RNA sequencing and library builds. It's not going to be 5 days. It's going to take some time to build libraries. I'm sure you would respect that. And So if you're doing nanopore sequencing, this is first come, first serve. We try to go as fast as we can, but it is our most popular sequencing service right now. And we have a lot of them going on at the same time. So keep that in mind. All that data for next-gen sequencing and nanopore is uploaded to the Advanced Computing Center. And I can tell you, uploading some of this data from nanopore can take a day. So banking of funds. This is something people ask us, can we pre-bill? And you can't pre-bill, but I will tell you, you can't bank funds with us. But if you start a project with us and you submit samples and we start doing quantitation, and it's like a 2-week process to get through, we can bill that under these special circumstances. And it might take a while, but one of the things I want to make sure of is that if we do this, we'll call it pre-billing. It's not really pre-billing. We want to wrap that billing up in 2 months for the sake of administrative processing for iLab. So don't come to us and say, I got $25,000, I want to put it in the core, because we don't do that. But we will bill it 2 months out if we need to. And of course, we're always willing to give letters of support for your grants. Please ask us. We want to give these to you. We want to be part of your grants. If you have like extra work that needs to be done and you want to see if you can pull in a technical person to put 5% on your grant, we're happy to be part of your project from a grant effort perspective.
Me too.
We all have something extra to add to this talk. But you guys outlined it well, Roxanne and Y and then Nicole. Please acknowledge our facility. And if you need to know how to write it, we will give that to you. But acknowledge our facility. We need that acknowledgement. We don't have something that's requiring it on our iLabs right now. But now that I've seen you guys present that, I think we might actually have that here. So. Okay, so I'm going to let Julie talk about bioinformatics, but thank you for your time.
Okay, last but not least, bioinformatics. So I'm Julie Dragon, I direct bioinformatics core. And we are a little bit of an outlier in the CBSR. We don't quite operate the same way. You can't just go in and request hours of our time. We participate in the consults with our colleagues in the genomics core, post-doctorate microscopy. And we like to know about your project in advance, talk about your biological question and what your needs are. And then we scope out the size of your project. We bill by the hour, not by the sample. We bill by the sample, but it's really about how much time it's going to take to deliver to you the analyses you need to answer your research questions, and it can differ with every project. So how do I get bioinformatics support? You need to reach out to one of us, and there's a bunch of us in this core. So myself, Ramiro Brontes, Emily Guswa, Princess Rodriguez, and Heather Driscoll. Heather Driscoll and Emily are members of the Vermont Biomedical Research Network that is dovetailed in with their data science core and our bioinformatics core, and work with us on a regular basis. And then in bioinformatics cores, our Princess and Ramiro. So you can set up a consult in iLabs, like everyone else has described, or you can just reach out to one of us directly to get the ball rolling on your project. And we encourage you to do this for anything, any question you have, even if it's not, doesn't turn out to be relevant or you don't need our support in the end. We're happy to answer questions and help you think through and troubleshoot your projects for you and your students. And generally our processes will participate in a consult depending on what you filled in your iLabs request, and we'll assign a bioinformatician who has the most experience or expertise in that particular domain to be part of that consult and probably ultimately work on your project. And also depends on who has effort available at the time. And you can also interact with us by attending courses or workshops. So Princess Rodriguez teaches 2 bioinformatics courses in the MD department. And we actually, as a group, offer workshops through the data carpentries, network carpentries program, also funded by the BBRN in Linux programming, R, GitHub, and yeah, those are the ones we've offered so far, but we're offering like 2 or 3 of those a year. Different levels of participation. So that's another way to interact with us. And actually Ramiro is managing a blog. We have a blog to look at. We always say we're going to post more content than we actually get done, but we're getting there. So what do we offer? This is often a question that I get, like, well, you haven't done this, or you don't do this, or I want this specific pipeline. So you guys know that bioinformatics is a continuously changing field. Constantly new things are coming on board. And we really don't shy away from any sort of analysis project. A lot of the parts of these analyses are all the same. And things sort of evolve as the software gets better. But it's really about stitching together a bunch of different processes to get your data from proteomics or genomics or whoever, and get it analyzed in a form that you can actually interpret, right? So we have free consultations. We are investing a lot of time in something called NF Cores, which is a Nextflow core workflow manager, and it's an effort in our community to standardize, if close to again, just standardizing analyses, because that's the reproducibility problems in bioinformatics, right? Because every software has a gazillion parameters you can tweak, and people often don't report those in the pubs that you read. So it's kind of an issue in our community, and this is a effort to get everybody on the same page. And the benefit of these NF cores is there's pre-written modules now, and they're loaded on the back, and they're actually pretty user-friendly to interact with, so we can help you or your students figure out how to run your data, like your genomics data, through one of these pipelines yourself. So we're happy to do that, or we can, participate sort of the soup to nuts level. People sometimes ask us just to do the pre-processing. I just need the fast views. I just need an alignment, or go through annotation and pathway analysis, or really get into model development. Depending on the research question, we can tweak models, rerun your data under different parameters to get you the best information out at the end. We also do training in R plotting, data mining, and we can build containers for running certain analyses over and over again. And we provide data management. You submit large amounts of data to the SRA and then document the work we've done. We don't post our outputs in iLabs as the other cores do, because they're usually too big. We create a web page. We send you a link to it. There are links in it to download your data from the back. A little bit different there. So yeah, we're getting close on time. So just reach out to us. Sometimes people say, well, I'm not sure how to get bioinformatics support. Just email any of us. You go to our webpage. We're happy to start a conversation with your project. Should I wrap up there?